So welcome everybody. We're going to have a great session today. This is a program that I'm really passionate about. It saves me a lot of time. So I'm excited to have Craig Zinda from NVIDIA come in and share his knowledge with us. He is has a background in HPC, working with TAC, and a background in bioinformatics. He comes from the plant side of things, which I won't hold against him. It's much more difficult than us with the, just a simple 2 sets of chromosomes. So, parachromosomes. So, I want to welcome Greg and hand it over to him, and welcome everybody else to this workshop on NVIDIA Parabricks.
Okay, and just so no one is scared, there will be a hands-on portion of this. So I'm just going to start with slides to get everyone introduced, and then we'll go hands-on. But yeah, so I'm Greg. I've been at NVIDIA for 3 years now on their healthcare team. And so I'm here to talk to you about accelerating genomic analysis with Parabricks. So the healthcare team at NVIDIA. So we do more than just gaming GPUs. These days, we're not only interested in like the awesome GPUs that are in the data center at Texas A&M, but also the applications that they can be used for. So we pay attention to 4 main categories in the healthcare team. So imaging. So that's everything from like medical imaging to like pathology as well, like whole slide analysis, medical devices at the edge, genomics, which is what I'm going to be talking about today, and then drug discovery. So that's everything from like traditional MD to, I guess, quantum, like quantum mechanics as well, and using AI for essentially synthesizing new drugs these days. So we've got a couple of platforms, well, toolkits in the NVIDIA Clara umbrella. So there's Manai, which is for medical imaging, Flare, which does federated learning. So the academic space is awesome and often unique, but where you can share data, or it's often expected for you to actually publish it publicly. But if you end up working with like a research hospital or a pharmaceutical company, it's a way for you to collaborate on the training of a deep learning model without actually having to transmit any data back and forth. There's NVIDIA Holoscan, so that's our edge medical device. So it's essentially like an ARM platform with a enterprise GPU inside it, and it's meant for essentially, I guess, augmented reality diagnoses, like in the operating room, just at the edge. Parabricks, which is accelerating everything genomics, and then BioNeMo, which is our framework and our framework for multi-GPU training of drug discovery models, and also an inference framework as well. And so these all are powered by our AI libraries and possibly NVIDIA Omniverse, and they all run across our hardware. So these days we do GPUs, we do DPUs, which is essentially one of our types of network cards. We make CPUs these days as well, and we have various, I guess, server platforms as well. There we go. So talking about how we interact with genomics. So we interact essentially with all 3 layers of genomics. I'm going to be focusing on the secondary, but I just kind of want to cover where we do interact with companies. So on the primary analysis side, you can think of this as taking place on the sequencer itself. So whenever you're processing a sample, if it's short reads or nanopore, oftentimes it's either images or like an actual signal that's going through the sequencer, and it classifies those signals into the actual nucleotides, the ACES, Gs, and Ts. And so a lot of sequencing companies, they work with us to accelerate that on the back end. So there could be NVIDIA tech inside your sequencer that's running like essentially TensorFlow, PyTorch, or TensorRT. They could also be running CUDA as well. But that's just literally base calling. So usually as soon as you get the reads, like the raw FASTQ files, that's when you move on to the secondary analysis. And so that's where you'd be doing, if it's a novel genome, you'd have to assemble it, or you'd align it to a known reference. And so that's where NVIDIA Parabricks comes in. So specifically on the alignment side, so we do GPU accelerated alignment, variant calling, and that's both for DNA and RNA. And if you've ever used Parabricks in the past, there used to be more tools, but essentially we decided to focus on fewer tools just to really focus on speed as well. Lastly, so if you have a background in bioinformatics like I do, a lot of the time, I mean, obviously like alignment does take time and calling variants does take time, but usually if you're working on a paper or a specific experiment, the actual like data science, like essentially aggregating those results and interpreting them using statistics, that always took the most amount of time for me. And so we have I'm going to dive into this a little bit at the end, and we have a platform called Rapids. So that's our essentially GPU accelerated version of scikit-learn, NumPy, NetworkX, anything else you want to do. It does a really good job of essentially, so if you've got a function you want to call on scikit-learn, instead of scikit-learn, import scikit-learn as SKL, you essentially just do import QML, and all the functions, they essentially have one-to-one calls. And it's just to essentially speed up those analyses. And there's a good use case of single cell analysis that I'll show at the end. Alrighty, so Parabricks. As I mentioned, Parabricks, it focuses on accelerating a few key applications. We're going to go into them, but essentially it's not meant to accelerate every single thing you can do with sequencing. It's essentially, if we, NVIDIA, if we wanted to make the largest impact in sequencing, what tools would we want to accelerate? So common ones you might know are GATK and BWA. And so we figured we'd have a huge impact. So we got up to 80x acceleration by moving the algorithms from CPU to GPU. These days, Parabricks is all containerized, and it's easy to call using popular workflow frameworks like WDL, Nextflow, and Cromwell. And we're essentially improving accuracy as well. And so the results for GATK, like haplotype caller, they're essentially the exact same as the CPU, but we also support things like deep variant, and that we actually make it easier to retrain the base deep variant model. And so you can actually fit it to your population. All right, so talking about the tools that Parabricks supports. On the alignment side, we've got BWA-MEM for aligning DNA reads and STAR for aligning RNA-seq. From the pure alignment, we sort the output, we mark duplicates, we do base quality score recalibration. We also, once those BAM files are generated, we collect metrics, and you can even convert those BAMs back to FASTQ, all GPU accelerated. And then on the variant calling side, we've got haplotype caller, which is from GATK. We've got deep variant. We've got MUTEC2 and Star Fusion for the RNA pipeline. So this is all meant to support population genomics. So cancer genomics, which usually focuses on human samples, but all of these, so I'm going to be doing, in the hands-on portion, I'm going to be using Arabidopsis just because I am a plant person, and that's usually the references I know best. And so all of these are generally applicable. You essentially just have to re-index whatever genome you want to work with, and it'll still be blazing fast. So to give you an idea of how fast, so on BWA-MEM, going from CPU to GPU, so CPU takes around 4 hours to do an alignment. I'll also illustrate this in our hands-on portion. It goes down to 11 minutes with BWA-MEM, and this is a 30x whole genome sample. So depending on your genome complexity and your sequencing depth, all these numbers will be relative to data size. On the actual like sorting of the BAM file, marking duplicates and doing base quality score recalibration, that goes from 9 hours to 6 minutes. Something that's always super duper impressive to me is the haplotype caller. So that goes from 16 hours down to 4 minutes, which is a huge speed up in my mind. A deep variant, that goes from 5 hours down to 11 minutes. And deep variant is actually pretty hard to run. If you've ever looked at their documentation, there's a whole step of like splitting out your, essentially sharding your data to support multiple GPUs. And Parabricks does that in an easy manner. So if this is a tool you use, I suggest you give it a try in Parabricks. And then Mutec 2 goes from 31 hours to 45 minutes. So huge. We're going to illustrate this hands-on, but just hoping taking a look at these numbers will inspire you to give it a try. I already mentioned that the latest, starting with Parabricks 4.0, we're now compatible with workflow managers. So we have templates to start from if you use Whittle, Nextflow, or Cromwell. So essentially, if you have a bioinformatics sequencing center or a lab where data is constantly coming in, or you need to download huge sets of data and you want to process it while as they complete downloading, this would be a good thing to check out. So yeah, essentially, these are more reproducible ways to run analyses instead of just having a single bash script that could get lost. This is more a way where you can template out the analyses you want to do for let's say every single set of DNA reads that comes in. We also offer enterprise support with Parabricks. So obviously there's a free version starting with 4.0 where you can download the container, run it. But if for some reason your sequencing center needs guarantees or SLAs for running these tools, we can offer that as well. We've also been making impact, huge impacts in the community with Parabricks. So it's been adopted by the Human Genome Center of Tokyo, where they're running it on 80 V100 GPUs in their compute cluster, just because the amount of time that they're saving for the reanalysis of these genomes makes sense. Regeneron, a UK biobank, they are reanalyzing almost 500,000 exomes in the UK biobank. And so Clara Parabricks makes that feasible instead of just slowly doing it as these, originally these were aligned as the data was slowly uploaded to biobank, but essentially they can do a realignment in just one batch by running Parabricks on GPUs. We also hit a world record with the Stanford University. So this was cool. I don't have a lot of I don't interact with a lot of clinical work too much, but this is a really cool story where there was an infant that had some sort of disease, and obviously it's hard to talk to the infant and figure out what's wrong, and so they decided that genotyping and figuring out what the disease was their best option. So they used an Oxford nanopore sequencer, and they used Parabricks for the actual alignment and variant calling. And so it went from sample collection to essentially diagnosis in, I think it was 7.5 hours. It was I want to say it was 7, but it might have been between 7 and 8, but essentially same day. And so, yeah, it was kind of cool to hear that they were able to diagnose this specific disease in one day instead of just, I don't know. Usually when I'm sick, I have to wait weeks for tests to come back sometimes. So getting started with Clara Parabricks. We have some slides here. These are going to be shared out at the end. There's some minimum requirements that I'm going to talk through here, but I'm going to hop over to my tutorial website and share that out with everyone. And then we can do that live. But overall, you need a GPU an NVIDIA GPU that supports starting with CUDA architecture 60 and at least 16 gigabytes of memory on that GPU. So common examples are T4s. However, with I think it was Parabricks 4.1.2, we actually added special support for Hopper, which is the H100s, and I know these are available on ACES. And so that would be worth testing out as well. I think today we're going to be using A30s, which obviously have enough memory, and they're plenty of powerful GPUs, but it could be worthwhile in the future. In addition to GPU memory, you also need CPU system memory as well. So these are guidelines for if you're using 2 GPUs to analyze a sample. We'd recommend around 100 gigs of memory, but to me that's overkill, especially for the samples that I'm using. I mean, anything from like 32 gigs of memory, it'll throw an error if you do run out of memory, and you can always try to allocate more if you're on ACES, because usually if you're doing SRun, I learned this the hard way, or I at least had to refamiliarize myself with the system yesterday, where you actually allocate memory in addition to the number of CPUs and GPUs. And you need a certain number of CPU cores to do some pre-processing on the data input. The heavy lifting is done by the GPU, but the CPUs are still either unzipping data or just prepping the reads for the GPU. You also need a modern driver. This recommends 465. I can't remember what the driver requirement is for 4.2, what we're going to be doing today, but I checked on ACES. They've got a modern driver and running the latest version of CUDA, so we're good. And I tested everything beforehand. And so where can you run Parabricks? This will run on NVIDIA DGX systems, any NVIDIA certified servers running with NVIDIA GPUs. You can run it in the cloud. So AWS, Google Cloud, anywhere with NVIDIA GPUs as well. If your sequencing center also uses things like Terra from Broad, DNA Nexus, or LifeBit, they also have some GPU accelerated pipelines that are running Parabricks on the back end, and you may not even be aware of it. So if it ever feels like an analysis is running really quickly, Parabricks is probably behind it. In addition to the workshop today, we have a Launchpad environment. So let's say if for some reason, I mean, everyone is on Access, and so you obviously have access to the ACES and maybe even the faster system. But if you ever need an environment to test out Parabricks, NVIDIA has these Launchpad labs also for other things besides Parabricks, so I recommend taking a look around. They're curated labs where essentially you can sometimes bring your own data, but data is always there, and you get an idea of how quickly the analysis can be done when it's accelerated with GPUs. And this is where I'm going to switch windows, so be patient with me. Alrighty, I'm going to put a URL in chat. And so this is what I'm going to be talking from. And this is where you'll be able to see the commands that I'm running. Just closed captioning is fine. Okay. Is my window visible for anyone, for everyone?
Yes.
Awesome. So while I'm rambling here, I already have a job running on ACES. Does everyone know? Is everyone logged into the system? Can everyone start up a job? So essentially, let's see. Is it P GPU? Yeah. Okay, so I'm the only one on here, so I think that's fine if I lose my job. But right now I'm on a login node. Everyone should try to get a job if they can. I think there's 96 CPUs and 6 GPUs. So I'm going to be saying I want one task, 16 CPUs for that task. I'm going overboard and saying, give me this job for 8 hours. I'm asking for an A30 GPU. I'm asking for 64 gigs of system memory. And there's also a reservation called training. And I want it to be an interactive session. So I'm doing --pty and then --l for a login shell.
Can you copy and paste that into the chat for them? And then
yes, very good idea.
Does anybody need instructions on how to get to this point right here? I have slides. I can show you how to. All right, Tatiana, this. Okay, let me just share real quick and get people to that point.
Yeah, that's good. I can
Okay.
stop sharing.
All right. So can everybody see my screen? The first step is just to go to hprc.tamu.edu. And once you're there, go over to the portal tab and click on ACES portal and then parentheses access. Once you have done that, you'll be asked to log in with your identity provider. And you should be able to do that. And then finally, that should get you on the landing page for our open on-demand portal. Once you are there, then you'll just go to the clusters tab, and that'll bring down the drop-down ACES shell access. And if you click on that, that will take you to the shell where you can log in with the command that Greg shared with us in chat. All right, I guess we're good to go. So I'll stop sharing.
Okay. Yeah, and then anyone can come off mute and ask questions anytime. I don't feel bad. Like I won't be upset. I'll be happy with any feedback or comments or questions. I'm hoping that they're allowed to come off mute.
Yes, I actually have one question. The CPUs that we have to use here are the same ones that are linked to our account in HPRC? Or because I'm understanding we're using ACES, I guess I'm a little bit confused because I know I have some of these ones in Terra and Grace, but I never used this one before. I don't know if I'm clear.
Are you talking about SUs?
Yes, SUs,
Oh,
I'm sorry.
Okay, no, yeah, you're on a training allocation, so you're not going to be charged anything for today.
And if I decided to run actually a job with the genomes I have, how should I proceed?
You would need to get an allocation through Access, which is really easy to do. It's just like it's not much different than getting an account through or an allocation through the HPRC.
Okay.
But you just have to go on to the Access website and fill out a form for an allocation. If you want to email me directly, I'll throw my email in and I'll help you with that process.
Awesome, thank you.
Yeah.
All right, I will get back to sharing my screen. Alrighty, and then if you do run the srun command, the way to know that you're actually in a job, it'll go from some like ACES login, there could be any other number besides 3 there, to the name of an actual compute node. So it'll be AC probably 64 if you run the exact command I did. All right. The first link that I shared, this is my little tiny workshop page for Parabricks. So the goal of this, so I'm going to go through some of the introduction, hopefully not reiterate too many things. But essentially, it's going to demonstrate how to align DNA reads with FQ to BAM, call variants with haplotype caller, and then go through some potential downstream analyses. So with the Arabidopsis, I've got a little tiny fun experiment if we have time for that we can do. But overall, this is just essentially just core functionality. So requirements, obviously you need a semi-modern GPU driver and an NVIDIA GPU to run Parabricks. Parabricks is containerized. So let's say if you have a workstation with an NVIDIA GPU with enough memory, you can use Docker to run the Parabricks container. For all the clusters at HPRC, I know that they support Singularity. Alternatively, there's Aptainer, which is the weird political fork of Singularity, and also Enroute. But essentially, it'll work with all container systems. In this, I'm going to be running just a single container in this in my little tiny environment that I'm going to have you all load up. But essentially, we've got the main Parabricks container. So we're going to be looking at 4.2 today. And technically, that does all the steps. However, if you need to re-index any of the references, you'll need the GATK and BWA containers. But since we're going to be reproducing everything that you can do with Parabricks on the CPU as well, just kind of illustrate those time differences, this is just a list of containers you can use to reproduce it in the future. I've also pulled all data for everyone, so you don't have to, we're not going to waste time downloading things, but essentially I pulled the just essentially 10 million reads from a paired end strain of TDR7 or Arabidopsis thaliana. That came from the 1001 Genomes project. I also pulled the TER10 genome. I removed the CHR chromosome names just to match the SNP, the conventions used by the 1001 Genomes project for actual variants. And I indexed it as well for everyone. And if anyone wants to reproduce this, I've got a little tiny script to download the data, but it's all been loaded up for everyone already, so you don't have to worry about it. So I've already done the intro to Parabricks. Feel free to refer back to it on your own, but I'm just going to skip over that.
Hey, Greg, if we have, will your tutorial work on the H100s?
Yes, it will, if there's any available.
Yeah, we have some in the reservation. So if you have, if there are users that haven't been able to get on the A30s, you can just remove.
So this, in the GRES argument, there's a GPU, right now it says A30. If you just switch that to H100, you should be able to get a different GPU. So
yeah, I can see that there are 5 or 6 people on the A30 node, but we do have H100 nodes that are reserved as well. So
if anyone wants to hop on an H100, I will be interested to know the time difference between the A30 and the H100. So even Wes, if that's you,
I'll do that.
I won't mind. So this container, as I've said, is free, and it's you can pull it from NGC. So with Docker, it's a Docker pull. With Singularity, it's a Singularity pull, and you'll get a CIF file at the end. And so what NGC is, this is our catalog for all the containers that we support. So this is the homepage. If you type Parabricks, there's an NVIDIA Parabricks container. So this will give you an overview of how to get started. It'll also give you a list of tags. 4.2.0 is the latest. That's what we're going to be going through today. But essentially, if you create an NGC account, you can see like security scanning of these containers because they're meant to be enterprise grade. You can see the layers, how they're built a little bit. And it gives you like a quick start for the user guides, for getting to the documentation for Parabricks and how to run it, obviously using Docker. So it's going to be a little bit different today. So as I've been saying, I have an environment that I've loaded up for everyone. So if you can copy these 2 commands here, so there's a module use and a module load. That's what I'm going to be talking through today. So I'm already on my compute node. I'm going to do a module use, and I'm going to do a module load. So at that point, so if you type ml avail at the top here, you can see there's a Parabricks workshop module and a snip match module. That's what's going to get loaded when you load the Parabricks workshop module. So at that point, you should have a PB run in your environment, and it's there. So this is actually calling Singularity in the background, and I know Wes has another module for Parabricks. I just wanted to use the latest one today, just because in the last couple of versions, they did add extra support for H100, so I wanted to make sure we had the latest and greatest going.
I'm
you can
getting
also,
I'm getting an error here.
oh, it's permissions. It's my fault. I'm
Yeah,
sorry.
I thought it might be. Okay.
I think you don't have to execute the Lua script. I think you just need read, because I didn't even have execute privileges on it. You might have to do the unuse and then use again. So you could do unuse and then use, and hopefully you don't get an error. And that's the only thing I can think of is maybe Elmod is. Oh, I know why. Try now. I think I had to open up permissions on my Scratch directory.
Got it.
Okay,
That was it.
That was it.
Yeah.
Sorry. I see some messages in the chat. I'm looking at them right now. Okay. And is it Yuchi? Were you able to get the module loaded after I just fixed that? Awesome. And Myra, are you good as well? Awesome. Perfect. Okay. It seems like everyone should be good now. Just in case, try the unuse and then use and then reload everything. And so just a note, if you end up using Singularity on your own in the future and you want to use GPUs, there is this --NV flag. And so I linked to the documentation. So if you're, and that essentially mounts the GPU devices and some libraries into the container at runtime. I'm doing that for you on the back end, so you don't have to worry about it. And so does Wes with his other module. But let's say you've been poking around NGC and you see that we have a PyTorch container that you want to run. I highly recommend, yeah, so that's how you essentially invoke that container with the --NV flag. Alrighty. So starting with DNA alignment. So there is so all these directories, I've set up a bunch of data and environment variables for everyone to use, so you don't have to, but essentially I've got 10 million reads, and this is a relatively small data set. So this is all to keep it very interactive, not only on the GPU side, but on the CPU side. I've got the indexed TER 10 reference, and it's indexed for BWA and GATK as well. And then I've got these, essentially it's like a variant table for 1001 genomes for various Arabidopsis types. And so that's the data we're going to be working with. If you do an ML show on the Parabricks workshop, you can see that I've populated a bunch of things for everyone. So it's more than just Parabricks. Okay, at that point, I recommend everyone go to the Scratch directory. And if I could type that out, that would be awesome. And so this is usually the file system that you want to do like any IO on. And so that's where I'm going to be putting like all my output files. I've already got a directory called work. That was the first word I came up with. And so that's where all my output's going to be during this workshop. So right now it's empty. So no one should need any files right now. I've got it all set up for you. And so usually whenever you start an analysis in bioinformatics, you're starting with FASTQ files, usually not a BAM, just because no one trusts anyone's alignments. And Parabricks makes it really quick to realign things. So feel free to just always download FASTQs and realign everything really quickly. But essentially when FASTQ to BAM is run, it goes through alignment. It sorts the output by coordinate, it marks any duplicates, and then it does base quality score recalibration sometimes, if you ask it to. And so we've got documentation as well. So before I dive into that, I just kind of want to look around the documentation. There's a link to it here. But I also want to just kind of go through our documentation page of, let's say you're starting with Parabricks, what are good things to look at? So the what's new is always worthwhile. So I think it was 4.1.1. No, it might have been 4.1. So with 4.1, they added support for Ada Lovelace and Hopper architecture. So they specifically added dynamic programming instructions to these generation of GPUs. And so that actually sped up sequence alignment. So I think 4.1 is on the system already, but always worthwhile looking to see what's new. In 4.2, we increased the version of GATK. So now we're compatible with GATK 4.3. Previously it was 4.2. And they increased the read length that they support as well. So that's like the big high level. In addition to that, so we've got guides on getting started, software overview. So these are all the software tools that's inside Parabricks and gives you an idea of what they do. We also have compatible CPU versions. I'm going to be going into this, but essentially if you need to reproduce any outputs produced by Parabricks, you can with just a CPU as well. So it's great for publications. We have guides on best performance. So specifically, if you want to squeeze out the most performance on a system, if you have enough GPU memory and it's a modern enough GPU, we've got options that essentially can accelerate things a little bit faster. So that's mostly for large data sets. With my toy data sets today, you may not see a huge difference. We've got tutorials. So there's a tutorial on running human genome data as well, all the way from FQ to BAM to calling variants. We've got a guide on doing deep variant training. I recommend checking out. Other how-tos, tools, things like that. All right. So sorry for the whirlwind there. I just wanted to cover it before I moved too far down the line. So FQ to BAM, it does have a lot of options that it supports. So obviously there's everything doing input. So it's either, this assumes paired end for NFQ. There's also single end. You can give it a list of FASTQ files. You can give it interval files, but lots of options, lots of BWA options as well. The things we're going to focus on are just the input reference. So it already has to be indexed. The in FASTQ files, what we want to name our output, which is our output BAM, and the number of GPUs to use. And so it's automatically detected. If anyone ran their job with multiple GPUs, you can choose between like the max or like 1, 2, 3, 4, whatever is on your system. So this is just a reminder to actually run it against a custom index or custom reference. You have to index it. I've already done it for you, so you don't have to worry about this. I've also populated these variables. So right now, all we're going to be doing is taking our input reads. So we've got, sorry, wrong window. If you remember, I'm also making some assumptions that everyone is familiar with sequencing. So this is our reference. This is the reference for Arabidopsis. It's been assembled. There's essentially five chromosomes and a mitochondria and a chloroplast genome sequence as well. Yes.
One quick question. I'm so sorry. I don't know if I'm the only one that I cannot access those files. I can move into the module files, but I don't have that directory or those files.
Yes, so you shouldn't. You're going to
Okay.
be, so the files are open for you to read, and so you're just going to be reading from them. So you should have you can do an echo ref, and then you can do an ls ref. And so you should be able to read that file from my environment. So this is just to keep everyone from having like 20 gigs of data like replicated around. So everyone's going to be sharing my data.
Okay, and also when I do the CD scratch, I cannot see those files or those directories.
Yep, nope, they're all in my environment.
Okay,
so
okay.
you're in your scratch and I'm in my scratch, but
Okay.
I've opened up my scratch so you can copy files from it.
Okay. Thank you so much.
Yeah, of course. All right, so we're going to do, I'm going to copy this command. And so what this does is I'm going to time it. So Parabricks does time sequences or time every step that it does. But I just like having that sanity check at the end of timing things. I'm going to do pb run fq to bam, which is we're going to align DNA to a reference. This is all helper variables now. So reference, it's pointing to our TER10 reference. This is just a variable, so no one has to type out these horrible paths. For the FASTQ inputs, we've got read 1 and read 2. And we've also got, this is the BAM that we want to create for output. And, oh, what did I do? It's just, I'm just going to repaste it. I don't know what happened. There we go. And I'm going to delete the num GPUs just because I only have one, so it doesn't matter. And I'm going to kick it off. And I want to do a sanity check. SSH. Okay, so it's working correctly. Just sanity checking things here. So it's going, it takes a second for the initial data to get loaded into the GPU, and then it goes pretty darn quickly. I can actually, so that's running over there. And if I do top, I can see that it's using CPU. I can also do NVIDIA SMI and see that it's using 43% of the A30 right now. So if I use commands like GPU write or GPU sort, it might use a little bit more of the GPU, but just to give everyone an idea. So this is, if you have a job with multiple GPUs, you can increase the number of GPUs, and you can see how that affects the runtime overall. So I totally expect everyone to be stuck at this stage right now. I'm just talking while that's happening. We also, so one of the cool things, I've mentioned it with Parabricks, is that we essentially re-implemented CPU tools. So BWA, GATK, these are open source tools that we essentially moved the algorithms to the GPU. And they are meant to reproduce the output of a specific version of each tool. So for FQ to BAM, they not only provide, they say that you need, where is it? So we've got the compatible CPU versions. Where is that over here? software overview, compatible CPU versions. You need BWA 0.7.15 to actually reproduce the output of FQ to BAM. And they also give compatible CPU-based commands. So if you ever want to sanity check the output, or if you want to publish a paper and you want to allow people to reproduce your results without having to own an NVIDIA GPU or access to an NVIDIA GPU, you can say, my results can also be reproduced by running BWA mem, using GATK to mark duplicates. And then if you want, you can do the base recalibration score as well. In my little example, we're not doing base recalibration. But to kind of like illustrate this, I wrote up a little tiny function called FQ to BAM PE, and this calls all the CPU versions of the tools. And I'm going to start that in my other window just to kind of to illustrate how long these steps take. Oh, I have to load my module. Sorry. There we go. And so this is all running on the CPU now. And we'll see how long that takes. I think it took 2 minutes and 43 seconds on the GPU. And so it's 1:23 right now, and we'll see how long we go. Okay, so I was gonna actually, after that's done running, we can actually compare the output just to sanity check that it's the exact same. But while that's going, I think that we can move on and to the haplotype section. So at the end of the day, you should just have to copy this one line, the single line, and then run it in your environment. And then you should have a single pb.bam file and index that gets created. Has anyone not been able to launch Parabricks yet with these inputs? Okay. So it looks like the CPU version is almost done on the right side of my terminal. It's calling SAMtools right now. Okay, so that took almost 8 minutes. So it was about a 4x difference between the GPU runtime and the CPU runtime. Okay, thanks, Bob. And that only becomes, that difference in runtime only becomes greater as your data size increases. As I've said, this is only 10 million reads. It's usually like millions of reads, or like hundreds of millions of reads, especially if you're dealing with like human data or like large plant genomes, like they're usually on a whole different scale. But I wanted to keep this real time for the CPU analysis side. So we've got, now I can go back to comparing the CPU versus the GPU output. So this is just easy ways to kind of take a look at the differences between the BAM files. So you can do, so you can see that the Parabricks BAM file, it's actually smaller than the one created by the CPU side. So you can see that there's that discrepancy that I captured on the workshop page as well. However, that's due to compression levels. If you do a flag stat on each of these, TDR 7. So we'll start with the Parabricks BAM. And I know that my SAMtools is wonky. Sorry for the library warnings. So, so primary mapped with properly paired. So I usually look at the properly paired reads. So 17,874 properly paired reads with the Parabricks version. Sorted, marked. And I'm going to do the same thing. And obviously you can go through every single read if you want. I'm just using flagstat to give these simple metrics. And so you can see the same number of properly paired reads from the CPU side that I did. So I'm the only one that ran the CPU side, at least I think. But essentially I'm just trying to show that the output is the exact same between the GPU tool and the CPU version of BWA. So trying to give you more reasons to try to adopt this, that's all. So if anyone wants to come back later and give this a try, you can try allocating a different number of GPUs in your job and seeing how that affects runtime. You can compare the speed up between CPU and GPU like I did. The inputs that we used were compressed. You can see if uncompressing the reads as an input gives you if that changes the runtime. And you can also view the reads with SAMTools view. You can take a look at the outputs, but I mean, you don't have to. But essentially that would just say like, are the reads in the exact same order? Not just is the total number present? So you can see how exactly the outputs match up. Okay. Has Parabricks FQ to BAM finished for everyone? I can only assume silence means everyone is done and waiting patiently. So I'm going to go through the haplotype caller. Yes. Okay, Wes is done. All right, so the other popular pipeline is haplotype caller. So this is the documentation for GATKs. And so one of the reasons I chose Arabidopsis is one, it's got a nice, tiny, small genome that's easy for real-time workshops, but it's also diploid, just like the human genome. So a lot of the tools, they just work right out of the box, and it's easy to understand. So similar to FQ to BAM, there's a bunch of options. We're just going to be running on defaults today. If you have experiments where you have you've got opinions about the way you want to run haplotype caller, there is probably a way to reproduce the exact arguments in GATK into Parabricks. But right now, we're just going to be focusing on our reference that was already indexed. So with GATK, so similar to like you have to SAMTools index or BWA index, there's a GATK create sequence dictionary, and then you have to do a SAMTools FAIDX on the reference. So that produces indices that GATK can use. You have to have an input BAM. So that's the output from FQ to BAM. You have to specify the output file. So this is your out variant file. So it usually ends in a .vcf. And so you can also specify the number of GPUs you want to use. I think the first time I did this workshop, I had 2 GPUs in my job, so I was playing around with that. So once again, to run haplotype caller, everyone ignore these first 2 lines because they don't work. Actually, the TDR BAM, that does work, but I've already populated it for you. So yeah, only copy this 1 line, and I can be the sanity check to make sure that works. Boom. And that works. And this is going to be quick. So that's just kicked off. Once again, we have a CPU equivalent for reproducing the haplotype caller. And this calls GATK haplotype caller on the back end. And I'm going to run that in my other window. And this one actually takes a really long time. So it's just going to be goofy. So this is, you can see that this gives a progress meter. We've already finished essentially chromosome 1. We're on chromosome 2. We're on chromosome 3, chromosome 4. On the CPU side, we're on chromosome 1 still on chromosome 1. So variant calling is like, this is the easiest way to illustrate like the difference in runtime of using the GPU or not GPU. So this is always the most fun on this workshop. So this took 1 minute, 11 seconds on a GPU. I have an out VCF file as well.
One
And
question.
So, yes.
What should we do if we need the outputs by chromosomes, for example?
So you could either pull out specific chromosomes from your BAM file because it's indexed and sorted. So it's easy to do like a SAM tools view. It's been a while since I've done this. View. So it does. Chromosome 1. If I remember correctly. Sorry. I'll just cheat here. So the region, that's it. So SAM tools view 1. So I don't have CHR1. I have 1, 2, 3. And so you can say, I want all the reads from chromosome 1. You can pipe it to a new file, pipe it to SAM tools view. Oh. There's like an argument for, essentially you can, I'm going to cheat. I'm just going to do like chr1.sam. And that's going to pull out all the reads that are aligned to chromosome 1 and put them into a SAM file. So that's about 1 gig.
You can use the I think it's the -B and the -O flag.
Yeah, that's the way you're supposed to do it. I can't. It's been a long time.
Yeah, I know.
With all the piping.
But yeah, it's just -BO and then 1. There's no piping. It's just the 1 call.
Oh, I see.
So SAM tools view, yeah.
-B
O, no, like lowercase o, and then you put the name of the chromosome, the BAM file there. So you don't even need to redirect it. Yeah.
Okay, let's see if that works.
I think that's.
Yeah, that's going to take a little bit longer because it does compress it. You can see on the variant caller side that we're still on chromosome 1, even though we've been done for a long time. So that would be a way to pull out. So that's 209 megs for the reads. That would be a way to pull out like a single chromosome worth of reads. And then you could call variants on just that. I can't remember if, let's take a look in the documentation to see if there's a way to specify a specific chromosome as well in this stage. Bam, Rico. Interval. So you can use L as well, and you can specify like an interval in this example, or you can do just a single chromosome of, in our case, it would be -L1. And
yeah, I have used this parallel L command before. on
Okay.
my pipeline. But after that step is where I get things messier because then I have to how is it called? Consolidate my files, and that's a thing that I really struggling in GATK. That's what I was asking.
When
but
you say
thank you.
consolidate, are you talking about like multiple samples that you
Yes.
want to compare? Okay, from all chromosome 1 on that sample.
On different samples, yes.
Yeah, okay. So that is more of like the downstream processing. And so
Mhm.
Parabricks will get you to that BCF as quickly as possible. And then you will have to use your data science skills to parse out those specific things and do those comparisons. And there's also like open source tools that'll do that.
Okay, I understand. So it will just give me the whole thing and then I will have to parse it myself.
Yes. And
Okay.
VCF files, similar to BAM, like they can be indexed as well. So you can produce the VCF for the entire genome, and you can pull out specific chromosomes as well.
Okay, great. Thank you.
Yeah. So CPU equivalent is still running. There's optional exercises after we're done here where there's a germline pipeline where if running FQ to BAM and then haplotype caller is a nuisance, you can run it in just one command called germline. So that's in our tools. Germline. There's a germline pipeline. So that does FQ to BAM and haplotype caller all in one. How much is the runtime affected by multiple GPUs when running GATK? We also have deep variant. Recommend you give it a try. We try to make it as easy as possible. You can also retrain deep variant. So the model that comes with deep variant is trained on human data. It's not trained for Arabidopsis. So you could always try retraining it on Arabidopsis data and seeing if that affects the result. We've got 15 minutes left, so I'm going to skip this. This takes a couple of minutes, but essentially the files that I downloaded, it had genotypes for various accessions of Arabidopsis. And the whole point was we were going to identify which variant our data came from. And I guess we could maybe do it. I'll just kick it off and see how long that takes. And so if you have a well-studied organism, you can do things like that. Optional exercises. And so I've got a presentation I'm going to dive back into, but you can try genotyping other samples. There's a web browser as well, or you can pull other data from 1001 genomes. You can try our RNA-seq pipeline. So the RNA FQ to BAM and then star fusion. We have deep learning institutes. So we have a DLI. So these are these are from NVIDIA. These are courses that you can take to learn more about our tools. So this DLI is for running deep variant, and the other one is variant calling on whole exome data. So it's going to be very similar to what we just did in this workshop, but the data will be exome data. Other things I want to mention are, so let's say you want to do more GPU accelerated bioinformatics. There's a bunch out there. So things that I wanted to mention are rapid single cell. I'm going to talk about that in the slides. I'll pull back up in a second. That's made possible with rapids. There's also something called Metacache GPU. So I also used to do metagenomics. And so this is a GPU accelerated way to classify reads. And so there's a tool called Metacache, but they also have a GPU version, and it's super duper quick. And if anyone does metagenomic analyses, you could say Kraken is super quick. The reason why Metacache is cool is because it builds. So Kraken is quick when you have a known reference database of like various like pathogens. If you need to modify that database, Kraken takes a long time and a lot of resources to rebuild that index. And Metacache does it really, really quickly on a GPU. So that is the main reason of why to use it. All right, I'm going to switch over to my slides real quick, because I know we're running out of time. Presentation, share. Alrighty, so where we left off. Ah, okay. Sorry, there's things. So I'm going to share out these slides, but there's just common little questions. You can use multiple GPUs, can't use multiple MIG instances. So that's relevant for A100 and H100. If MIG is enabled, that cuts the GPU into little bits. Parabricks does not go multi-node, so multi-GPU only, but that works well for, I know it worked well for faster. Is ACES also a liquid system?
Yeah,
Wes?
we're working on it will have where you can compose multiple GPUs to a node, and the end goal is that users won't even be able to tell that it's being composed. So you could just go in and ask for however many GPUs you want, and all that gets sorted out on the back end. So hopefully sometime within the next year, that'll all be sorted out. So
nice. Yeah. And then the other thing is when it says we need 16 gigs of GPU RAM, it means that RAM needs to be free. So you can't be running like PyTorch or anything like 2 runs of Parabricks on the same GPU. So just be aware of that if things start failing. So the last thing I wanted to like inspire everyone with is to, if you do single cell analyses, this is becoming more and more popular with even spatial becoming more and more relevant. Wanted to talk about software for that. So similar to how do you support, you were talking about comparing the BCS from multiple samples. With single cell, they're comparing gene counts across millions of cells these days. So this obviously this timeline ends in 2017. So there's even like 30 million cell data sets where it's like 60,000 genes or 30,000 genes by however many cells as well. And so not only is that a hard problem from just like a system memory standpoint, but also processing that data is difficult and clustering it. So we have rapids, and what that does is it takes like common steps from like using pandas, scikit-learn, networkx, TensorFlow, matplotlib, and just bringing all those tools to the GPU. So we've got cuDF, which is a one-to-one implementation of pandas, cuML, which is scikit-learn, and it's all on it. Every time you do an operation of data getting loaded into memory, it's actually going into GPU memory with something called Apache Arrow. And it's keeping all that data in GPU memory to make it as quick as possible for any further processing steps. And so to give you an idea of the difference, so normally for pandas, it's import pandas as pd.read CSV. You can do import cuDF, and then it's the same read CSV function, and it just goes into the GPU. And so average speedups, if you use cuDF for processing data files, 150x. For scikit-learn versus cuML, 50x speedup, 250x speedup for cuGraph if you do network analytics with networkx. So highly recommend you check it out, just like Parabricks, but this is more general purpose data science. So this is just XGBoost showing that it scales. So up to 100 CPU nodes versus just 16 GPUs, how much faster. But the thing I wanted to show off here is for single cell analyses. So as I was mentioning, it's usually you're looking at these gene count tables where you're analyzing like 60,000 genes by like millions of cells, and it's a huge table. And so there's this whole data processing stage of filter normalizing the data, do dimensionality reduction with PCA, visualization, clustering with k-means, Louvain, Layden. And it all takes serious amounts of time. And so not only is it faster on a GPU, but if you're running in the cloud, it's also cheaper just because it goes from like 5 hours down to 11 minutes. So you can also see the speedups for each of these steps. So we've got an interactive notebook for playing with this, and you can actually like highlight little bits of data and then rerun the whole like clustering step. And it's real-time interactive. Obviously, loading all the data does take time, so you don't see the most speedups there. But as soon as you get into like the clustering, where we're seeing like 3000x faster with running on a GPU versus a CPU, Louvain clustering, like overall the pipeline is 27 times faster if you run it on a A100 GPU, and using 64 CPU cores. So if anyone's doing this, highly recommend checking it out. In the workshop website, there's a link to Rapid Single Cell, and that is so we've got a little tiny notebook here, but Rapid Single Cell is, we worked with a lab and they are maintaining our GPU accelerated pipelines now. And so that works on top of ScanPy, if anyone's familiar with that. So, and then just resources, this is all in the workshop website, how to get started with Rapids. But I think that's it. And so if anyone wants to ask questions, feel free. If you want to come on camera and introduce yourself and share about your research, I'd be happy to listen as well. Yeah, thanks for attending today.
All right. Well, thank you very much, Greg.
Yeah.
I guess if anybody wants to stick around and talk, we can hang on for a few minutes. If not, we really appreciate you guys coming today.